Here you can see a concerned customer and one of our epidemiologists providing some insight. Let's take a closer look.
You seem confused looking at the peanut butter.
Well, I heard something on the news about foodborne pathogen outbreak. Do you know anything about this?
I could probably provide a little insight, actually. I'm an epidemiologist.
Epidemiologist? What is that?
So, epidemiology is the study of the distribution and determinants of disease, and the application of this study to the control of diseases. So, as an epidemiologist, I work with an entire team that investigates foodborne outbreaks.
Oh, that's really neat. Tell me more. Should I be concerned?
Fortunately, there are no ongoing outbreaks associated with peanut butter, but if you are interested, I can tell you more.
Yeah, I would really appreciate that.
The CDC defines 7 key steps to investigating an outbreak. The first is detecting a potential outbreak when multiple individuals are reported being sick and having similar symptoms, so you need at least 2 for it to be an outbreak. The second step is to start collecting more info to find more related cases that got sick within the same time frame. The third step is beginning to hypothesize possible sources of contamination, similar to what you heard on the news about the peanut butter. Steps 4 and 5 are testing and then confirming the hypotheses of food sources of possible contamination. At this point, it is time for step 6, which is controlling the outbreak. This relies on a variety of options and usually includes recalling the product. The last step is determining that an outbreak is over when the number of cases decreases to a normal, expected level. Do you want to learn how we use epidemiology and the genetics to track foodborne pathogens causing an outbreak?
Yes, if you have time.
Definitely, follow me.
Hi. Good morning. How are you?
Pretty good. Not really good.
I heard you're sick.
Yes, I'm not feeling well.
Here, one of our epidemiologists has begun asking questions to an individual who has experienced symptoms related to our foodborne outbreak. The Pennsylvania Department of Health has a survey questionnaire that is broken into 7 distinct parts that help identify possible sources for foodborne pathogen outbreaks. Those include Part 1, demographics, Part 2, clinical and dietary information, Part 3, events, travel, or restaurants that may be relevant, and Part 4 is an open-ended food history of the last several weeks. Part 5 is restaurant exposures and the foods you may have had at said restaurants. Part 6 would be other food exposures, and Part 7 finally would be any animal contact that may have occurred in the last couple weeks. Let's listen in some more to hear some of the questions that our epidemiologist may ask.
Any ethnic or specialty markets?
No, I don't think so.
Okay. Any fresh eggs?
Fresh eggs? Cooked eggs.
Cooked eggs?
Yeah.
Okay. Any specific type of meat or poultry?
Beef.
No? Beef? Okay.
Chicken.
Okay. Good. Prepared foods. Have you had any peanut butter in the past?
Oh yeah, peanut butter. I had peanut butter every morning last week.
Oh wow.
I love peanut butter.
Oh wow, every morning, okay. And any specific brand or?
I can't really remember,
Okay.
but I can go back and check.
Okay, perfect, yeah. What about any fresh fruits, vegetables, zucchini, eggplant?
No.
No?
Like apples, oranges?
Apple? Yeah, okay. Perfect. Awesome. Sounds good. I think we're pretty much done here.
Okay.
Any further questions, we'll contact you.
Okay, thank
Okay,
you.
perfect.
CDC defines epidemiology as the study of distribution and determinants of health-related states among specific populations and the application of that study to control the health problems. Epidemiology is a discipline which can be applied to many areas of public health, whether it be the study of cancer, obesity, or in our case, foodborne outbreaks. Anything that the CDC refers to as a health-related state. For an outbreak to occur, there must be multiple related cases occurring within a limited time period. A single, unrelated case is referred to as sporadic and is not part of an outbreak. Usually, outbreaks are identified by observing an increase in incidences of a disease in a particular group of people. In the case of foodborne illnesses, epidemiology enables investigative agencies to determine the source of disease-causing pathogens or toxins. The ultimate goal of an epidemiological investigation related to a foodborne illness is to prevent the continued spread of the illness and to retroactively provide information to regulatory agencies to craft legislation which works to protect against future outbreaks.
Once we generate a hypothesis and identify potential food item that is likely contaminated and has caused outbreak, scientists collect suspected food samples and conduct lab testing to isolate disease-causing bacteria. After that, they determine if the DNA of the bacteria isolated from patients matches with the DNA of bacteria isolated from food or the environment in which the food was processed. POSNet is a national laboratory network that connects foodborne illness cases to detect outbreaks. It uses DNA fingerprinting and whole genome sequencing of bacteria, which make people sick and detect local and multi-state outbreaks. Whole genome sequencing is the analysis of the entire genomic DNA sequence, and it can be achieved through the sequencing by synthesis. Let's watch a video from Illumina that describes the process of sequencing by synthesis.
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with alon, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification.
In conclusion, according to researchers from the Department of Health in Pennsylvania, there are 6 steps of laboratory protocol: collecting the suspicious foods, lab testing, whole genome sequencing of collected bacterial isolates, sharing the data with CDC, data analysis, and submission of data to NCBI database.
The PulseNet program and whole genome technology allows us to do much more than just match human and food isolates. For example, it allows us to predict whether the isolates will be resistant to specific antibiotics. Antibiotic resistance has become one of the most pressing issues in public health today. The Pennsylvania Department of Health is using isolates from foodborne outbreaks to also study antimicrobial resistance among pathogens. If a specific Salmonella strain that causes the outbreak is resistant to certain antibiotics, then treatment of infected patients will not be as effective if those specific antibiotics are used. That is why it is so important to understand antimicrobial resistance in foodborne pathogens, and the Department of Health works closely with the National Antimicrobial Resistance Monitoring System, also known as NARMS, to monitor antimicrobial resistance in Salmonella and other pathogenic bacteria. NARMS has 3 facets: testing samples from foodborne illness cases, as we would do with the Salmonella outbreak, testing meats for sale at a grocery store, and testing livestock samples. The first facet is reactive, while the other 2 methods are more proactive measures. Antimicrobial resistance can be thought of as the tragedy of the commons. A microbiologist from the DOH described this as when everyone has access to a limited common resource, it is often squandered. This manifests itself as the overuse of antibiotics and by patients not completing their full rounds of antibiotics. Both of these lead to decreased efficacy. Now, there are some pathogens that have become resistant to nearly all known antibiotic classes. This is why the CDC refers to antimicrobial resistance as one of the biggest public health challenges of our time, as each year 23,000 people die from an antibiotic-resistant infection. The technologies you just previously heard about can help to reduce this number. The Department of Health does more than even just antimicrobial resistance studies. Using these technologies and different microbial techniques, they also can look at the evolution of virulence, meaning how pathogenic or how intense is a certain outbreak, and they also do work on bioterrorism, specifically anthrax and how it might be used for bad purposes. The DOH does all of this and investigates along every step of an outbreak, all within their one building.
No. Now that I learned all about outbreak investigation and epidemiology, I know that the peanut butter is probably safe to eat. I learned about the 7 steps of outbreak investigations, the basic epidemiological principles, about whole genome sequencing and other cool technology today. I also learned about antimicrobial resistance and about the work being done every day by the Department of Health and epidemiologists just like you.
Well, I hope today was insightful for you and you're no longer concerned. If you have any more questions, feel free to contact me.